So today I want us to continue with the manufacturing basics we started with last week. And last week we were focusing more on tableting technology. And we looked at the various steps that are involved in tableting. We went on to have a look at the tableting cycle or the compression cycle. And then we went on to have a look at the problems that you can encounter during tableting, and made a suggestion of how you can handle each of those problems. Then we also went on to have a look at the coating process. That's if you want to proceed and do coating, of which you've got a number of reasons why you would want to do that. Today, I want us to focus on manufacturing of Microcapsules and also Microcapsules. We won't go a lot more in detail with microencapsulation, but we'll mention them, and then we'll get to look at this a bit more in detail when we start doing modern pharmaceutics in 5th year. So we want to have a look at characteristics of capsules as dosage forms, manufacturing of hard gelatin capsules, hard gelatin capsule filling, the various filling methods that are there. Then we'll look at gelatin, soft gelatin capsules, and then end up with microcapsules. So capsules are solid dosage forms with hard or soft gels. So in this case, we see that the categorization would depend on the type of the shell that is being used. So if you have the hard shell, that would be referred to as the hard gelatin capsules. And if you have got the soft gelatin, then that would be the soft gelatin capsules. For capsules, we see that you've got various shapes and sizes, and these might contain either a single dose of one or more active ingredients. So depending on the design, you are looking at ingredient, or you can have more doses and more of active ingredients. Naturally, we see that these are intended for oral administration. Of course, the technology has allowed for development of other capsules which can be administered by other means. So in capsules, the drug is enclosed within a hard or soft shell. These capsule shells are made of gelatin or other substances, the consistency of which may be modified by the addition of substances such as glycerol sorbitol. So these substances which are added to modify the consistency, we see that these are considered as plasticizers, and plasticizers, we know that they are able to modify the flexibility of the materials or the polymers, in this case the polymer being gelatin. So once you are able to modify flexibility, you can shape them in a manner you would want them to be. The shell should disintegrate in the presence of digestive fluids so that the contents are released. So inside the capsules, you have the active ingredient. You could have other excipients, but once this shell gets into presence of digestive fluids, you want the shell to disintegrate so that you can release the contents. Okay, so one of the consideration is that those contents should not cause deterioration of the shell, because that is going to affect the stability of the dosage form and can then reduce the shelf life. So the excipients for capsule production, we see that we have almost all the excipients that we looked at under tableting. Even in capsule technology, you can find all these. So you've got the diluents, the solvents, excuse me. You've got the surface active substances. You've got the antimicrobial agents. If you need a preservative, you can have sweeteners, coloring matter, you can have flavoring substances, disintegrating substances, glidants, lubricants, substances capable of modifying the behavior of the active ingredients in the GI. So we have all these excipients available for selection, and you can pick depending on the characteristics of the capsule you are looking for. Remember, we had mentioned when we are looking at tableting that the starting point is actually the same for these two types of dosage forms. So you can start with weighing, you do the dispensing, you do the screening if you have to do milling to reduce the particle size. You can do all those things. If you are dealing with the powders which do not have good flow characteristics, you can even embark on granulation. So you produce free-flowing granules, and then instead of now going to tableting, you take those granules, you lubricate them, and then you can pack them into capsule shells. So the starting points are similar. Categories of capsules, we have different categories. We've already mentioned we've got hard capsules and soft capsules. That is depending on the strength of the shell that you are using, or the flexibility of the shells you are using. And then you have what are referred to as modified release capsules. So these modified release capsules can either be one of these. They can be hard capsules, they can be soft capsules. These are also then subdivided into delayed release capsules. You have gastric resistant enteric capsules, and then you have sustained release capsules. In the first one, this is where you are delaying the release of the contents of the capsule. So you pick a polymer that is capable of resisting gastric fluids, so it remains intact in the stomach, and there are various reasons why you'd want to do that. Maybe the drug is sensitive to acid. Okay, acid labile degrades in acidic media. So to prevent its degradation, you can design it as an enteric capsule, enteric drug delivery system, so that it is only disintegrating once it evades the acid medium. Then sustained release capsules in cases where you want to prolong the release of the drug. Okay, and there could be reasons. Mostly when you have a sustained release product, you want to simply make it convenient for the patient to take this, and also ensure that you've got uniform drug concentration in the blood. So for hard gelatin capsules, classified as a solid dosage form, the drug is enclosed within the hard shell. The shell is typically made up of gelatin. Gelatin is one of the polymers you looked at in third year. They are primarily intended for oral drug delivery. Gelatin disintegrates very quickly in GI contents, and so there is rapid release of the drug. So if you look at gelatin itself, gelatin is prepared by hydrolysis from collagen, which is from animal bones and skin. It is classified into two. It's got you've got type A, which is derived from pig skin, and then you've got type B, derived from animal bones. It's got a number of properties, essential properties, what that makes it very useful. These include stable when dry, so its stability ensures that the entire drug delivery system is able to stay for a prolonged period of time without degradation. And then subject to microbial degradation when moist. So we see that as far as storage is concerned, at all points you have to avoid moisture because that can encourage microbial degradation. This at times, although it might appear like a disadvantage, this can also be used as a mechanism of drug delivery. Okay, so you can use the ability of gelatin to be degraded by normal flora as a way of drug delivery. So you allow for degradation by the normal flora, normal microorganisms that are present in the GIT. And that will result in release of capsule contents. So it could be one way of designing a sustained release product depending on microorganisms. Then the other characteristic is that it is easily digested. Okay, so that easy digestion of gelatin also does contribute to drug release, but also most importantly, it means that you do not have capsule shells lying around within the GIT, so it can be easily digested and absorbed. Okay, another characteristic, and it does undergo absorption. So this ensures that you have this capsule shell removed from the GIT. So hard gelatin capsules mostly typically will contain 12 to 16% moisture. They are not devoid of moisture, but they are also typically filled with dry solids. So you can fill in the capsule capsule shells. You can have powders, you can have granules, you can have pellets, you can even have tablets. Okay, there are cases where a tablet is placed inside of a capsule shell. Okay. And then they also contain the colorant and the preservatives, like we indicated when we listed all those excipients that you can find. So there are different sizes and shapes of capsules that you can have. So here in this table, you have different sizes and the indicative volume here. So we see that the smaller the indicated size, the larger the volume of the capsule. So you've got what we call 000 size. You've got 1.37 cubic centimeters. Then you have 00 capsule size, 0.95. You've got 0.68. And as the number of the size increases, the volume decreases. Some of the advantages that hard gelatin capsules have: tasteless and odorless. So for drug substances that have bitter taste or unpleasant taste and odor, you can use hard gelatin capsules. Swallowing is easy. A lot of people find it easier to swallow capsules compared to tablets. Flexibility in formulation. We see this flexibility, it's coming from the fact that you can easily vary the amount of powders or granules or pellets that you are going to incorporate in these capsule shells. Uniquely suitable for blinded clinical trials. We see when we are talking about clinical trials, you can simply pick a certain capsule shape or capsule size of the same color, and it will be easier for you to conduct blinded clinical trials. This is in a case where the appearance of all the capsules that will contain the active ingredient is the same as that which do not contain an active ingredient. So you will not be able to tell which one has got API and which one doesn't have. And so it makes it easier to prepare such samples for clinical trials compared to tableting. Useful for extemporaneous compounding by pharmacists. So these are quite easy to prepare. So you can easily fill in the capsules if you decide you are going to extemporaneously prepare capsules. It's easier for you to simply weigh the powders or granules and then you fill in the capsules. This extemporaneous compounding is manufacturing on a small scale. Also, the other way, if you want to use already filled in capsules to produce, for example, suspensions, you can easily simply open the capsules and use the contents to prepare suspensions. That happens quite a lot in practice in cases where you find that a particular medicine is only existing in capsule form or tableting form. You can, and then you need to prepare something for maybe an elderly or a pediatric patient who cannot swallow the solid dosage forms. Some of the disadvantages tend to be more expensive to produce than the tablets. Okay. So first you have to produce the gelatin shells. That's an added step. So it tends to be a bit more expensive compared to tableting technology. Not suitable for highly soluble salts. We know that because there is a presence of moisture, we had indicated that there's about 12 to 16% moisture. highly soluble salts will be able to dissolve. And once they dissolve, that might become problematic. The stability of the entire dosage form is going to be affected. So for highly soluble salts, hard gelatin capsules are not useful. So ideally, we see that hard capsules have shells consisting of two prefabricated cylindrical sections that fit together. One end of which section is rounded and closed, and the other is opened. So every one of us have seen how capsules are made up of. So you've got two components. So one component is actually referred to as the capsule head, and then the other one is referred to as the main body of the capsule, or you could call it a tail. So these two are fitted together whilst enclosing the material in them. The contents of hard capsules are usually in solid form, powders or granules. And then you have soft gelatin capsules. Soft gelatin capsules are hermetically sealed one-piece capsule containing a liquid or semi-solid fluid. So we see that whilst in hard gelatin capsules, you've got two components. In soft gelatin capsules, these are hermetical one sealed piece. So you appear to simply have one piece, okay? So soft capsules usually formed, filled and sealed in one single operation. So whilst in production of hard gelatin capsules, you can have a number of steps that ranges from production of the gel and then production of the shells themselves. And then you fill the capsules with whatever content you want, and then you close them. Production of soft gelatin capsules is done in a single step. So within a single step, you've got formation of the gelatin capsule, and then there's filling that's taking place, and then you seal in a single operation. So advantages of soft gelatin capsules, these show improved bioavailability as the drug is presented in a solubilized form. We've seen that in soft gelatin capsules, you have mostly liquids or semi-solid. There's also enhanced drug stability, the protection that is conferred upon the contents by the soft gelatin, and also considering there are fewer steps in the manufacturing, single step, that allows you to have a much more stable consumer preference regarding ease of swallowing. Soft gelatin capsules preferred by most patients. They are easy to swallow, so they give that convenience. As a result, you can improve the stability of patient compliance. Then offer opportunities for product differentiation via color. So you can produce different colors for this. So you've got an easy way of differentiating between these products. The soft gels can be enteric for delayed release. I think when we talked about the third type of capsules, where we mentioned, even here, you have an opportunity to have enteric coated soft gelatin cap. So in the manufacturing of capsules, you have a number of measures that you have to consider. Active ingredients, when present in solid state, form have appropriate solid state properties such as particle size and forms. So these are the same considerations that we make when we are talking about tablet technology. So particle size distribution, because this is going to be affecting the flow characteristics. So anything that affects the flow characteristics is going to also affect the ease of packing those samples into the capsules. Then issues to do with polymorphs. You want to ensure that all of them are existing in a similar solid state. We know that different polymorphs present with different physical properties, and we did talk about this under preformulation. So ensure that mixing with excipients is carried out in a manner that ensures homogeneity. You don't want the powders or the granules that you are packaging to be showing lack of homogeneity. So similar consideration here as well. So minimize the degradation of the active ingredients. Okay, so if you are going to be dealing with drugs which are sensitive to moisture, you have to take the necessary precautions. Minimize the risk of microbial contamination. Here you are also talking about semi-solid fuse or liquid fuse that you put in soft gelatin capsules. Then you also have to avoid cross-contamination. So there are various stages of production depending on which type of capsule you are dealing with. So if you are dealing with the hard gelatin capsule, you have these steps. You first of all prepare the gelatin solution, and then from the solution, you then prepare the gelatin shells. And then you prepare, once you have the gelatin shells, you have then to fill those shells. So there's hard gelatin capsule filling. Once you have them back, you are done with filling, you go on to packaging and labeling. Then if you are dealing with the soft gelatin capsule, you have preparation of gelatin solutions. But we see here, we had indicated that the process of producing the capsules as well as the filling happen within the single step if you are dealing with the soft gelatin capsules. So preparation and filling of soft gelatin capsules is done in a single step, and then you go to packaging and labeling. So you have one less step when you are talking about soft gelatin capsules. So there are different methods of obtaining gelatin solutions. So you can have the gelatin capsules by involving one stage of swelling, and in the other type, you have no stage of swelling. So when we talk about this, I think it's a bit more clear when we describe each of these stages a bit more in detail. So if you are going to have swelling, first, the swelling of gelatin in cold water. So you put gelatin in cold water, which is between 15 to 18 degrees Celsius. And you do so from 1.5 to 2 hours. Okay, so the gelatin is going to swell. So once it swells, that being one of its characteristics when it is exposed to water, then you dissolve the gelatin. Okay, gelatin then is dissolved. Then you can add preservatives, plasticizers, and other excipients. So For stability purposes, you can add preservatives. For improvement of flexibility, you can add plasticizers. Okay, and then you can remove any air bubbles from the solution of the gelatin by vacuum if there are any bubbles. Then you stabilize the solution at 45 to 60 degrees Celsius. Obviously, this being done here, we see that this is a higher temperature. You want to ensure that the solution can easily flow. Okay, that allows you to mold that solution into different shapes. Then without the stage of swelling, here you start by heating of purified water that is 70 to 75 degrees Celsius. And then you dissolve the preservatives and the plasticizers and other excipients that you think might be useful. Could be colorants, could be anything you want to add. Then you add to this water, and these are going to dissolve. And then to that, you are going to add gelatin. So gelatin will then dissolve at this high temperature. So it's not being given, but it's. And then you remove the air bubbles from the solution of the gelatin by vacuum again, and you stabilize the solution of the gelatin at 45 to 60 degrees Celsius. So you move from a higher temperature and get to this lower temperature, but still high enough for the flow of the solution to take place. Okay, so this is simply showing a gelatin melting tank where these processes we are describing are undertaken. And then you have methods of soft gel production. So in the manufacturing of soft gels, you have that are considered. You have what is referred to as the droplet method, and then you have the pressing method. Okay, so remember, we've just looked at how you can produce the gelatin solution. So once you have the gelatin solution, and then you want to produce soft gels, or soft gelatin capsules, you have two options you can follow. One is the droplet, and the other one is the pressing. Let's have a look at the droplet method to start with. So you have an apparatus that is in a simplified form designed in this manner. So here you have number 2 here is the hopper with medicine substance. So here you place the medicines, okay, that you want to fill in the soft gelatin capsule. Then here, number 1 here is the container with medicine substance. So we see that these are acting as reservoirs, okay, and then they feed the medicines into this container here. And then you have number 3, this is 3. This is the container with the gelatin solution. So you put your gelatin solution in this. Okay, so you've got pairs here and there. Okay, and then you have what is referred to as the conical nozzle. This one here, number 4. Okay, and then you have what is referred to as the pulsator, which is here. As the name suggests, this creates a pulse, pulses Okay, and then you have for what is referred to as the cooler. Okay, so the cooler is the whole lot of this number 6 here, this container here. Okay, and then you have number 7, which is the container with Vaseline oil. So the way this works is that once the apparatus is in operation, the medicines are going to be transferred to here, and then it will start flowing into this this tube. Excuse me. So in this tube, as it flows there, this pulse is going to be creating pulses, or like vibrating. So what is happening here? You've got the medicine flowing through, and then you have the soft gelatin. So the soft this gelatin solution. So this gelatin solution is going to be more like enclosing. In between, you've got the medicine solution. Outside, you've got the gelatin. The pulse is created